everybody to a new Global IMMUNOTALK. It's noon here in New Haven. And just briefly to remind you, if this is the first time you join us, we have generated this series with the goal of benefiting and inspiring immunologists across the world in an egalitarian manner and to increase the opportunities for scientific learning without traveling. Of course, we could not do this without the fantastic speakers that have agreed to present. We have today Erika Pearce, who will soon be introduced by my co-organizer, Elina Zuniga. And just to remind you that next week we will have Gabriel Rabinovich joining us. So with this, I will now turn it to Elina, who will introduce our global immuno speaker today.
Thank you so much, Carly. It is a true honor and a great pleasure to be introducing to you today Dr. Erika Pearce as a global immune speaker. So Erika is originally from US, although she now lives in Germany. But she obtained her PhD in 2005 at the University of Pennsylvania in Philadelphia, where she studied the regulation of T-cell responses during infection. Then she performed a postdoctoral study at the same University of Pennsylvania, where she began then her research into how cellular metabolic processes govern immune responses to infection and also cancer. In 2009, Erika launched her independent research career at the Trudeau Institute in New York. And in 2011, she moved her laboratory to the Department of Pathology and Immunology at Washington University School of Medicine in St. Louis. And finally, in 2015, she moved abroad to Freiburg, Germany, where she became a director at the Max Planck Institute of Immunobiology and Epigenetics, and is where she now resides. So as I am sure most of you know, Erika has pioneered the work of cellular metabolism in immune cells, particularly T cells. And she has made many terrific contributions that push this field forward, including, for example, the discovery of how mitochondria and the morphology of the mitochondria and metabolic pathways engaged by T cells can influence the development of effector and memory T cells. Erika's team also demonstrated that metabolic enzymes can not only regulate T cells by participating in metabolic pathways, but also affect translation of messenger RNAs encoding cytokines and thereby are able to post-transcriptionally regulate the T cell function. And so these and more of the really amazing breakthroughs that Erika's team has contributed have opened the exciting possibility that targeting metabolic pathways or specific metabolic enzymes in T cells could allow their therapeutic manipulation to improve their functions in the context of infections or cancer. So as you can imagine, based on her research, Erika has a long list of publications in top-ranked journals, and not surprisingly has obtained many prestigious awards for her work on immune cell metabolism. Among them, I would like to just mention that in 2018, she was awarded the Leibniz Prize, which is the most prestigious research finding prize in Germany. And again, this is one of many of the honors that she has received. So Erika, we cannot be more honored and pleased to have you here as a global immuno speaker. And we truly appreciate your efforts to share your science with us today and with this global forum that we have.
Well, thank you very much for that really, really kind introduction, Lena. Thank you to Carla for taking the effort to organize, and bring science around the globe. It's really nice to be here, and thanks to all the participants who are going to tune into my talk.
Thank you so much, Erika. And before we move to your talk, we always like to know a little bit more about our speakers by asking a question. And the question that we thought attendees would be curious to note from such a successful and a female scientist like you, Erika, is what is the trait in your personality that helped you the most in your scientific career?
I think it's being persistent, but tenacious, even in the face of criticism. You know, you just take it and keep going.
That's amazing. Yeah, persistence with what you believe in science. Yes, thank you so much. I'm happy to hear your thoughts. Okay, so if you wish, we cannot wait to hear about your research. So if you'd like to share your screen. Let's see. Perfect, we can see it perfectly well. Yeah, thank you.
Okay. Thank you. So today I'm going to talk about polyamine metabolism and how we found that it's important for regulating T cell lineage fidelity and the function of these cells. So what are polyamines? Polyamines are these molecules down here, putrescine, spermidine, and spermine. And polyamines can be made from the urea cycle, where arginine can be converted to ornithine, and then ornithine can be converted to putrescine via the action of ornithine to carboxylase, this enzyme here. And then once putresine's made, that can be turned into spermidine or spermine, and then back to spermidine. So polyamines are synthesized by metabolically active cells, but they also can be acquired from the environment because we get them from our diet, and gut microbes can also make polyamines. So there's a lot known about polyamines. This is just shown sort of an overview of what these molecules have been shown to do, taken from a review. There's many known functions, and you can see they're implicated in cell proliferation and gene regulation. Here you can see that polyamines can be acquired from the outside, but as I said, they can also be synthesized within the cell, and implicated in apoptosis and autophagy. And also polyamines participate in a very specific post-translational modification of a protein called EIF5A. And this modification is called hyperusination. And this can impact translation in cells. So what exactly is EIF5A? This is a messenger RNA and ribosome binding protein that is important for translation. And hypusin, which is this modification shown here, so this conserved lysine in EIF5A, and one enzyme called DHPS will take spermidine, it's polyamine, and through its action will make deoxyhypusin. And then a second enzyme called DOHH will make hypusin. And so obviously this hypusination of EIF5A is spermidine dependent. And what's really cool about this this entire system here is that EIF5A is the only known protein that contains hypusin. And the only known actions of these two enzymes are to affect this post-translational modification of EIF5A. So earlier last year, this is a picture of Dan Puliston, who's a postdoc in my lab, and Dan published a paper last year in Cell Metabolism showing that this polyamine hypusin axis has selective effects on the expression of a subset of mitochondrial proteins. And the work that he did in this was MEFs and macrophages. And Dan really brought this project to the lab, and he's thought about this for a lot of years. And he wrote a Wellcome Trust Fellowship to really pursue what this metabolic pathway is doing in immune cells. So as I said, he was able to show, that these polyamines and hypusin could have selective effects on protein expression, in particular proteins that were in the mitochondria. And this would have consequences for these macrophages and whether or not they can become alternatively versus assumed classical activation, and certainly the function of these cells. And the reason I'm mentioning this now is that I'm going to come back to this at the very end. But the story I'm going to tell you about today is in T cells. So for T cells, polyamine biosynthesis is engaged after activation. So I put this schematic here just to remind you of the pathway. Again, ODC, rate-limiting enzyme for the production of polyamines. And you can see that after T cell activation, either a day or 2 days later, ODC is increased in these cells. And looking here now at the production of putrescine, spermidine, and spermine, you can see that with T cell activation, these polyamines accumulate. So one of the fun things about working with T cells is this aspect of CD4 T cells that can differentiate into distinct subsets. And of course, we can do this very effectively in vitro, where you can isolate naive CD4 T cells and polarize them into, for the purpose of this talk, I'm just going to talk about Th1, Th2, Th17, and Treg cells using a variety of cytokine cocktails. And of course, these different T cell helper cells here will assume distinct functions through perhaps cytokines that they make, shown here for Th1 cells like interferon gamma, but they will also express lineage-specific transcription factors. And for a Th1 cell, that would be TBEG, and of course for Th2 cells, GATA3, and so on. And what's nice about this in vitro system that can be measured by these canonical transcription factors as well as cytokines that these cells make is that this differentiation of these CD4 T cells is important for a variety of protection against pathogens as well as mediating things like autoimmunity. So of course, Th17 cells are implicated in immunity. Things like Th1 cells are important for intracellular pathogens. Th2 cells for extracellular parasites, and of course Tregs are important for regulating these effector populations. So I just wanted to show now the polyamine enzyme levels that occur in these CD4 T cell subsets. So again, just looking at the differentiated T cells here, and you can see these polyamine synthesis enzymes. They're all expressed, maybe less so in Th17 cells, but basically they're all there. And this is just looking at the same thing by flow cytometry. You can see that ODC and spermine synthase are expressed in these cells across the board. And then if we actually look at the flux through the polyamine pathway, looking at heavy labeled arginine and glutamine, which can be used to produce polyamines in these cells. If you can trace heavy labeled arginine into putrescine, you can see that Th1 cells and Th2 cells pull a lot of that substrate into putrescine, whereas maybe Th17 cells do this to a much lower degree. Perhaps the same for spermidine. And so there's different movement through the pathway. But even though we see this different movement through the pathway, really, for example, you can take spermidine here, regardless of that difference perhaps in enzyme expression or substrate incorporation, the polyamine pool size is actually maintained across subsets. So thinking about that to then dissect further, what is the importance of polyamine biosynthesis in these cells? Since we know that they make a lot of them, it's turned on with activation. What is the role and why is it important in these cells? So to get at this question, one of the first things that Dan did when he came to the lab was generate these ODC flox mice and then cross them to CD4 Cre to generate mice deficient for ODC in their T cells. And then if you take these ODC deficient T cells and you look at expression of the enzyme, you can see it is indeed gone. And then if you look at what happens to polyamine synthesis, again, ODC, rate limiting step here, you can see that the polyamines that follow are no longer produced in an ODC knockout, as you would expect. So these cells don't produce polyamines after activation. So then we can look at what happens to the function that they would assume by putting them in vitro and differentiating them into a variety of these subsets. So as I mentioned before, when you polarize Th1 cells, this is just an example. Now we're looking by flow cytometry at interferon gamma and IL-17. So a T cell culture polarized toward Th1 will make a lot of interferon gamma and not IL-17. And Th2 cells, of course, won't make either one of these. Th17 cells will make IL-17, and Treg cells don't really make either cytokine to any great degree either. But when you look at what happens in the ODC knockout T cells, you can see that cytokine expression is really dysregulated. So not only do Th1 cells really pump up the amount of interferon gamma they make, you start to see things like Th2 cells make interferon gamma, as well as Tregs and Th17 cells. And even within the Th17 lineage, things like IL-17 production decreases. So there's quite a severe dysregulation, and you can see this across the subsets. Interferon gamma is up, and then you can see things like Th2 cells start to make IL-17, and you also get a lot of double producers. And of course, this is not just for interferon gamma and IL-17, but you can even start to see things like IL-5 and IL-13 be expressed within the Th1 subset, and then even cells start to double produce interferon gamma and IL-13 across the subsets, which arguably T cells should never produce both of these cytokines at once. And of course, this is not just cytokines when we look, but we can also see dysregulation of the lineage-specific transcription factors. So here we're just looking at TBET, which of course is a Th1 transcription factor. You can see that's high in these cells. Now I'm completely in the dark. There we go. Lost my train of thought. There we go. Okay, so this is increased in Th1 cells, and in your normal conditions in wild type cells, it shouldn't be expressed in these other subsets. But now when we look at ODC knockout T cells, you can see that Th2 cells start to express a lot of TBET, as do Tregs. And again, we're just looking at across the subsets, you can see pretty severe cytokine transcription factor expression across all of the different subsets. So then we wanted to know, okay, so if ornithine decarboxylase should be producing putrescine, could we put putrescine on the cells and rescue the phenotype? So we're showing here ODC knockout Th17 cells, as I showed you before, a wild type cell really makes much more IL-17. This wild type's making a little bit of interferon, but when ODC is knocked out, you get this big increase in interferon and a drop in IL-17. And now when we put putrescine back on these cells, it completely rescues the phenotype. So that was good. That showed us that the pathway was working as we assumed it should be. And this is just showing the same is true for transcription factors across the subsets. So unlike ODC knockout T cells in vitro, the mice that we generated, the T cell specific, mice that were deleted in ODC specifically in their T cells, they look completely normal. And we wouldn't have suspected, looking at what was happening in these T cells in vitro, that perhaps these mice should look normal. But we also knew that the mice could still obtain polyamines from the diet as well as gut microbes. And we reasoned that perhaps the amount of polyamines that they were able to obtain from those two sources was enough in a steady state condition. So we wondered, could we push the inflammatory phenotype by increasing the demand for polyamines in vivo? So to do this, we used an adoptive transfer model of IBD, where we could sort naive T cells, both from ODC and wild type, ODC knockout and wild type mice. And then we could adoptively transfer these sorted cells into RAG knockouts. And in the wild type condition, you'd expect to see IBD in about 4 to 10 weeks. We could see what would happen with the mice that received the ODC knockout T cells. And then we could, of course, sacrifice later and assess immune infiltrates in the organs. So one thing that we noticed is that unlike the kinetic for the wild type T cell transfer, which actually takes some time to cause disease in mice, the mice that received the ODC knockout T cells got sick very quickly. And began to drop weight precipitously. Their colons were thickened and red very quickly at this early time point, which is evidence of inflammation. And if we take T cells from the colon, you can see in comparison to wild type, these ODC knockout T cells in the colon are making a huge amount of interferon gamma, and they're actually making less IL-17. So really recapitulating the phenotype that we would see in vitro. So we could invoke an inflammatory phenotype by increasing the demand for polyamines in vivo. So next thing we wanted to know was how do polyamines actually direct this appropriate lineage specification? And some of these different aspects of what polyamines do could be difficult to test, but one that we knew that we could test very clearly was it the role of polyamines in hyperusination, and was this leading to the effects that we were seeing? Was that the important mechanistic role? So to do that, we decided to go back to these ODC knockout T cell model, and here we're showing ODC knockout Th17 cells, and as I told you before, the knockouts have really crazy cytokine production, but when you add putrescine back, it completely rescues the phenotype and they look like wild type again. So we could ask the question, you know, does this rescue still occur if the hypusine pathway is deleted in these ODC knockout T cells? So to do that, Dan took a CRISPR approach and deleted DHPS in these ODC knockout T cells. You can see here DHPS, when he introduces the guides for DHPS, the ODC knockouts no longer express that enzyme. When that enzyme is gone, you no longer can hypusinate EIF5A. We see this decreased in the wild type as well, and perhaps a decrease in EIF5A hypusin, but I think that maybe the deletion is a little less efficient here. These ODC knockouts have no hypusinated EIF5A. And then you can see here that putrescine no longer corrects that aberrant interferon gamma and Tbet expression in these knockout T cells when DHPS is deleted. So here are the ODC knockouts making a lot of interferon gamma, and these happen to be Tregs here, which shouldn't be making this cytokine. And then when you add putrescine, they go back way back down, so they look much more like normal Tregs. But now, when you delete out DHPS and you add back putrescine, you no longer get that rescue of suppressing interferon gamma production, and the same is for their aberrant Tbet expression. And we can also look at this in the other way, where feeding back putrescine to the ODC knockout T cells would normally restore IL-17 expression. Give back putrescine and they start to make normal levels of IL-17 again. But if you delete THPS, it doesn't completely restore that anymore. So what these data showed us was that the polyamine-mediated effects that we were seeing in this context were really going through hypusine, and that implicated a dominant role for the hypusine pathway in T cell lineage fidelity, and really tied the observation that the importance for polyamines in these cells is for, suggested that it would be for hypusine. So to actually test that hypothesis and really see if that was the case, was the role of these polyamines going through hypusine by providing substrate and spermidine to DOHH and DHPS. If we look in these cells, you can see the hypusine pathway goes up with activation, and the ODC knockout cells don't have as much hypusinated EIF5A. So that sort of fit the hypothesis. And then to test that very clearly, we collaborated with Stefan Balapanov from the University of Zurich, where we used his DOHH flox mice to generate mice that had a T cell specific deletion in DOHH. You can see here, they efficiently are deleted for DOHH, and you can see that hypusinated EIF5A goes down. And then when we look at what happens to the T cells upon in vitro polarization, we can see that they have dysregulated cytokine production, much like we saw for the ODC knockout T cells. Again, here's Th2 cells, no DOHH, they start making interferon gamma and IL-17. IL-17 production goes down, but only within the Th17 lineage, and in fact, things like Tregs start to produce IL-17. And here, this is just showing the dysregulation across the subsets. And much like for the ODC knockouts, this isn't just restricted to cytokines. If we look at the transcription factor expression in these cells, you can also see that it is dysregulated, much like it was for the ODC knockout T cells. Just one example, you can see GATA3 now being expressed in Th17 cells that don't have DOHH. But unlike what we saw for the ODC knockout T cell mice in the steady state, these mice that were deleted for DOHH in their T cells, they developed fatal inflammatory disease and die between 12 and 20 weeks. And this is characterized by increased interferon gamma as well as other cytokines in the serum. You can see enhanced IL-6 here, for example. And when you look at the colons, you can see intestinal thickening, increased infiltrates in these mice, shortened colons. So clearly they're also affected by colitis and dying of this. And if you look in the small intestine or colon, the T cells that are sitting in these tissue sites have dysregulated cytokines, much like what we saw for the ODC in the IBD transfer model. So that was pretty intriguing to us, because that in our minds was now really suggesting that the polyamine biosynthesis was all about hypusin. And if this were true, if the mechanism by which polyamines were regulating TH differentiation was through hypusin, then if we did a third model of DHPS deficiency, we should be able to see exactly the same phenotype as we saw for the ODC knockouts and the DOHH knockouts. So made one final mouse model here, again with Stefan Balabanov's mice, took the DHPS flocks and crossed them to CD4-CRE to make a T cell specific deletion. You can see these mice, they have neither DHPS and they no longer hypusinate EIF5A. And without going into it, their phenotype looks exactly like the other two models I showed you. So extreme cytokine dysregulation and transcription factor expression when DOHH is gone. Much like the DOHH mice, these mice also die between 12 and 20 weeks after developing platelet inflammatory disease. And again, their T cells that are sitting in these tissue sites have dysregulated cytokine production. So our conclusion from those data is that together, those three genetic models really tie this polyamine hypusin axis. And they show that polyamines are directing T cell lineage fidelity through the function of DOHH and DHPS. And again, the only ascribed function of these enzymes has been to hypusinate EIF5A. So next thing we wanted to focus a little bit more on how this was happening. Why does this axis, how does it act to focus lineage commitment and really confer appropriate lineage specification on T cells? So because we know that cytokine and effector programs are really patterned in these cells, and of course can be affected widely by chromatin accessibility, we wanted to assess these cells by ataxia to look at differential peaks of chromatin accessibility within the ODC knockouts, the DOHH knockouts in comparison to wild type cells. And what you can see here, we polarized all the different subsets, and the different symbols denote the different T helper subset. And the color is the genotype. And basically you can see that the ODC knockouts and the DOHH knockouts have a lot of differentially regulated chromatin accessibility. Some of the peaks overlap, some do not. But just for example, there's 10,000 differential peaks in these TH2 cells in comparison to wild type. So really widely altered chromatin accessibility. And as one might expect, that altered chromatin accessibility would also lead to differential gene expression. And if we look at these do the same type of analysis now looking by RNA-seq, you can see really widely altered gene expression patterns in these T cells. And these differential chromatin accessibility and gene expression were linked to broad changes in histone methylation and acetylation. So here we can look at a variety of epigenetic markers. Here's H3K9 acetyl and H3K27 acetyl across all the subsets, both for ODC and for DOHH knockouts. You can see significant increases in this acetylation mark. We can see H3K4 trimethyl, the same sort of picture here, and even things like a repressive mark like this H3K9 trimethyl, that's decreased in these cells. So broad changes of both histone methylation and acetylation. So we wonder, how do these really marked differences manifest? So because we were focused on T cells and we were seeing this prevalent interferon gamma expression across the subsets, wasn't the only thing that was up, but we thought, well, we should really consider TVET expression, which is also really highly expressed across all the different conditions that I showed you before in the hypusin polyamine knockouts. So TBET's known to bind DNA, of course, and it can also associate with chromatin modifying enzymes. And in that way, it can influence the epigenetic environment of the cell. So not act just as a transcription factor, but also influence the epigenetic environment in these T cells. So we wondered, is the increased expression of TBET actually responsible for driving the expression of the effector T cell genes in either the ODC, the DHPS, and the DOHH knockout T cells? We knew that the dysregulation went beyond interferon gamma, so we didn't know if that was going to completely correct these cells, but we were really curious to see if it would at least correct interferon gamma production. So what Dan did is again used a CRISPR approach to delete TBET in the TH1 cells in the ODC knockout background. So what you can see here is that you get an efficient deletion of TBET, that's no problem in these cells. And then when we look at the cells that have been, where the control guides have been introduced, you can see there's very little effect. Wild type cells make a lot of interferon gamma, but you can see increased interferon gamma and TBET expression in these ODC knockout T cells. Now, when you delete out TBET, you can see with the TBET guides, TBET expression goes down in both. And as you would expect, the interferon gamma production from the wild type T cells is really blunted now with TBET gone, which is what should happen, but it doesn't really seem to affect at all the interferon gamma production that's being driven in the ODC knockout. So we knew that the chromatin dysregulation, at least that was leading to interferon gamma, was not corrected by TBET deletion. It didn't even bring down the interferon gamma production in those ODC knockout T cells. And that really suggested to us a really profound defect in the chromatin state. So then we wondered, could major changes in the expression of chromatin modifiers, since there was so much, really highly gene dysregulation in these cells, could that contribute to the phenotype that we were seeing in these polyamine Hyperusination deficient T cells? So we looked at the RNA-seq and we focused on chromatin modifying genes, and we could see that there was significant changes in the expression of many of these chromatin modifiers, and they were shared between both ODC knockouts and DOHH knockouts. So with that in mind, we wondered, could the deletion of these chromatin modifying enzymes return polyamine Hyperusination deficient T cells back to a normal baseline functional state? So we took an approach to CRISPR a variety of the HAT enzymes in these cells. So one of the first things that Dan focused on was P300, which mediates acetylation on H3 and on H4. And if you remember, we saw increased H3K27 acetylation in these cells. And here we're focusing on TH2 cells because we're going to look at interferon gamma and TBET expression. Of course, TH2 cells really shouldn't be making either interferon gamma or TBET. And these histograms now are showing the results from the CRISPR experiment looking at P300 expression. So you can look at cells that have the control guides, wild type T cells in comparison to DOHH knockout. Right here, you can start to see this increased P300 expression, which would correlate with our RNA-seq data that suggested this, dysregulation in chromatin modifying enzymes. So increase P300, and now introduce the P300 CRISPR guides, and you knock down P300. So what happens to interferon gamma and TBET expression? You can see here that when you introduce the guides against P300, you can correct some of that aberrant interferon gamma production as well as TBET expression in these TH2 cells. So we went on, and this is actually showing the same type of experiment, but now looking at P300 deletion in the ODC knockout, TH1 and Tregs. And you can see for ODC knockouts here, if you delete out P300, you can correct their increased interferon gamma expression, both in TH1 cells and just showing Tregs here as well for TBET. Focusing on another HAT, we're looking at CAT2A, which mediates H3K9 acetylation, which was also increased by our RNA-seq. So this is just showing that this is deleted out efficiently in these cells. And again, with that, you can bring back these cytokine and transcription factors back to baseline normal levels. And the same is true. This is showing TAP1, another HAT. which again brings back the dysregulated cytokines and transcription factors back to baseline. So what this data was telling us is that their hats were really important and somehow contributing to the phenotype, but we wondered, is it really only the hats, or is it the acetylation state that's underlying the dysregulation? And of course, hats require substrates, and they require acetyl-CoA to acetylate histones. So ATP citrate lyase is an enzyme that produces acetyl-CoA, and of course, this is important for histone acetylation. And that enzyme can provide a direct link between metabolism and epigenetics. So we wanted to focus on a CRISPR-mediated deletion of ACLY in these ODC knockout T cells, shown here. We get efficient deletion. And you can see that ACLY deletion also corrects the aberrant interferon gamma and T-beta expression in ODC knockout T cells across the different subsets. Just shown here, brings down interferon gamma, as well in Th2 and Th17 cells, drops T-beta expression. So I think these data, the ACLY deletion and HAT deletions, implicate the acetylation state as the key underlying event that's driving dysregulation in the polyamine hypuzin deficient T cells. So we feel like we can really clearly make that case for acetylation. But if you remember in the beginning, when I talked about a paper that Dan had published last year looking in macrophages, and looking at this pathway really implicated the selective effects on the expression of many proteins that were mitochondrial proteins. And what the highlight of that paper really was this altered mitochondrial state that we were able to see when you would inhibit this pathway. And this is just showing some data, just showing that a lot of this is going to proteomics. You can see mitochondrial proteins are very much affected when you inhibit the polyamine hypuzin pathway. Many of the enzymes that are expressed in the mitochondria, their protein expression drops when you inhibit this pathway. And this is just showing by metabolite analysis, a very altered metabolic profile for TCA cycle metabolites when hypuzin pathway is inhibited. So acute inhibition of this pathway led to a selective decrease in the expression of many of these mitochondrial proteins and TCA cycle enzymes. So we wondered, could altered TCA cycle metabolites lead to altered substrate availability and also directly contribute to the altered epigenetic state? And we think this is certainly the case for acetyl-CoA, and perhaps these effects on the mitochondria, this is sort of what we're focusing on next. Or how you get this, altered state, because there's different substrate availability coming from this altered mitochondria state. So when we look at just looking at TCA cycle metabolites that are in these T cells from all three of the genetic models, you can see that TCA cycle metabolites are really dysregulated across all of the genotypes. So really showing how something very profound is going on with the mitochondria. And it's not only those TCA cycle metabolites, but if we look at 2-HG, which is an increased focus on this metabolite because it can function as an antagonist of alpha-ketoglutarate dependent enzymes and modulate histone methylation and chromatin accessibility. We see really dysregulated 2-HG. It's way down in these cells. And because we saw altered histone methylation, we wondered if this could also be contributing to that altered chromatin state. So to test that, I'm just going to show you one last piece of data here. We actually treated the cells with 2-HG. And in this experiment, there's also the C646, which is a HAT inhibitor. So that would mimic more of the CRISPR data of the HATs that I showed you before. And we're looking at DOHH knockout Th2 cells, which shouldn't be making lots of interferon gamma, but they do, as I've showed you. And when you add the HAT inhibitor, that drops interferon gamma. We're looking at T-beta here, sorry. We'll go to interferon gamma. Drops interferon gamma down. And when you add 2-HG, that also seems to have an effect to dampen that dysregulated cytokine expression, as well as T-beta expression. And you can see this as well for the double producers. They're inhibited in with either one of those treatments. And I think what is perhaps really intriguing as well and telling something very specific about what's happening with these epigenetic marks in the altered chromatin state is it doesn't seem to have any effect on the appropriate lineage specification. So where GATA3 should be expressed by these cells, and it is, any of these treatments doesn't seem to affect that GATA3 expression. It only seems to inhibit the diseased or the aberrant expression of cytokines and transcription factors. So that's sort of where we're focusing on for future work. So in summary, we think that this polyamine hypofusine pathway is really important for regulating T helper cell lineage fidelity. So we now know, we think with this study that polyamine biosynthesis in these cells to confer that lineage specificity is really critical to provide spermidine for DHPS and VOHH, because through the function of EIF5A, which could affect the mitochondria directly, and through that actually alter available metabolites through acetyl-CoA or perhaps through other metabolites, and then directly affect histone acetylation or methylation and chromatin accessibility. This also could have differential effects on just the transcription of chromatin modifiers and factors. Either way, this pathway has really profound effects on the transcriptional program that's enacted in these cells and is critical for maintaining clear T helper subset specification. So to acknowledge the people that had a hand in this work, really, this is all the work of Dan Fuliston, who's a postdoc in my lab and has been with me for a few years. The lab works together, so all these people listed here have provided support. I'd also like to call out David Sanin, who's in Ed Pearce's lab. He helps a lot with all the bioinformatics analysis that we do. And to just acknowledge the facilities at MPI, in particular proteomics and metabolomics, as well as the funding from the Max Planck Society. So thank you very much for your attention.
Thank you, Erika. That was terrific. Always learning so much from you. Yeah, and the depth of the and rigor with the genetic approaches in vitro and in vivo. Yeah, thank you so much for sharing that. I appreciate it. And so I think Carla will direct questions now to Twitter.
Yes, thank you, Erika, so much for your presentation. That was great. So for those of you that have questions, this is just a reminder. Please, you can go to Twitter. Oops, sorry about that. Oh, okay, went the other way. I'm sorry. Okay. You can go to Twitter and please go to the Global IMMUNOTALK account where you will find already a tweet that says ask questions for Dr. Erika Pearce here. You can reply to that tweet and just please remember to include the hashtag of Global Immuno. And today Erika is going to be using our ImmunoSpeaker account, so you can also include the @ImmunoSpeaker. So thanks so much, everybody, and thank you, Erika, and we look forward to seeing you next week.
Thank you both very much for having me. Bye, everyone. Thank you.
Thank you again. Bye.